Buenas tardes todos, bienvenidos otra vez a los Coloquios IB. Hoy tenemos el gusto de presentar al profesor Gilmore, que es invitado del programa Maldacena. Bien, les paso ahora el micrófono a Laura para que nos cuente un poco sobre él.
I will introduce Professor Ian Gilmore. He's a fellow of the Academy of Medical Science in UK and a senior fellow at the National Physical Laboratory. He also serves as a visiting professor at the University of Nottingham, and he is the founding director of the National Center of Excellence in Mass Spectrometry Imaging. With over 30 years of experience in mass spectrometry of surfaces and more than 160 peer-reviewed publications, Professor Gilmore have made groundbreaking contributions to the development of high-resolution molecular imaging techniques. And he developed the OrbiSIMS technology that has revolutionary the surface chemical analysis. He has received several prestigious awards, and to name a few, the Alfred Beninghofer Prize, the Acacia Award, and the UFTA Prize for Technology. And Professor Gilmore have also played a key role in international standardization in surface analysis, and he is serving on the UK Committee on Research Integrity. So we welcome Professor Hill.
Muchas gracias, Laura. And buenas tardes a todos. It's a great pleasure to be here. It's my first trip to Argentina. I've been very excited to come. And I'd particularly like to thank Laura for all she's done organizing the trip and the Maldacena program. And I'd also like to particularly say a big thank you to Horacio and to Andrea and to Liliana for everything they've done to make my week very nice, and very pleased. Mi nuevos amigos. Gracias. Okay, so I'm going to talk to you today about the work that I do in my group, and a technology that we developed called OrbiSIMS, and I'm going to give you some of the applications of that in material science and in life sciences. Most of my research is actually in the life sciences, but we also do work in material science, so I give some examples of that. I'm very often inspired by a famous biologist, a Nobel Prize winner, Sidney Brenner. He's famous for mRNA and things like that. And he says here, the big lesson to learn here is that in science, mathematics is the art of the pure, physics is the art of the optimal, and biology is the art of whatever wins survives. But another thing I like very much about this is that actually in the life sciences particularly, we need to work with mathematicians, physicists, and biologists together. And it's only by working together that we can solve these really difficult problems. And in my group, we have a real cross-section of different people all working together. And I think that's a fantastic thing. There's something I've seen in this institute as I've been through the week, is just what a wonderful environment it is and how collaborative you all are. And I think that's a real strength in everything you do. So if we're going to do analytical chemistry, there are different ways that we can do this. We can use light, of course, and this is very nice because it's non-damaging. We can look at the sample for a long time. We can do things in vivo quite easily. So examples are techniques like Raman and infrared spectroscopy. Or we can use the magnetic field, and then you have techniques like nuclear magnetic resonance or MRI, for example. Very, very powerful technique, really, for giving the exact identity of what a molecule is and what its structure is. And then we can use the electric or magnetic field to separate ions based on their mass to charge, to mass spectrometry. And with mass spectrometry, a nice advantage of mass spectrometry is, again, we've got very good analytical information. It's also a very sensitive technique, much more sensitive than NMR. And this is obviously going to be the focus of my talk in mass spectrometry. But my wife is a quantum theorist, so at home we're always discussing about quantum things, and this kind of led me on to this analogy. So you will all know in quantum mechanics the Heisenberg uncertainty principle. So the more certain we are about an object's position, x, then the less certain we are about its momentum, p. This is very well known, of course. Well, I have an analogy to this, which I call the molecular uncertainty principle. Is the more certain we are about a molecule's position, x, the less certain we are about its identity. And this is a general fundamental problem across all of industry and academia. We can take techniques like NMR, which will tell you exactly what the molecule is. We have very high confidence, we know what the molecule is, but we have no idea where it is. Or we can techniques like electron microscopy, which give us fantastic information about where something is, but we don't have, for molecules, for example, we don't have the molecular information to know what it is. So it's the kind of the work of my group to try and find what's the limit, how far can we push that limit to get absolute information about the identity of the molecule. And for example, if you're working in a drug company, we can't just kind of think, well, we think it's that molecule, because you're going to make $1 billion decision on whether you cancel a drug or you let it carry on to the clinical trials. You need to know, yes, it is absolutely that drug molecule, 100% confident. But not only do we need to know what it is, we need to know exactly where it is within the cell. And this is very difficult. So mass spectrometry imaging, how does it work? It's a very simple principle. We take a sample. It can be any solid state sample. In this case, it's a piece of tissue. And then we use an ionization source as a probe. And these probes can be photons using laser beams. It's a technique called MALDI. They can be chemical probes, which is a technique called electrospray. And then we can use ion beams. Ion beams have the advantage that we can focus them down. And I'll show you the kind of the works that we can do. It's a very high resolution. But in all cases, we address the sample in an array with a computer. And every time it hits the sample, we desorb molecules into the gas phase. Some fraction of those molecules are ionized, and it's only the ionized ones that we can detect in a mass spectrometer. Then there are lots of different kinds of mass spectrometers that we can use, and then we collect the mass spectrum for every single pixel. So you can see we now have a very large data set in our computer, and then we can decide to look at this particular molecule here, and then we can look at spatial distribution on an intensity map, or this peak, etc. And in fact, in the work that I'm going to show you, we can do by depth as well. So we can do an X and Y image, and then we can go down in depth to get a 4-dimensional data set. So this is very powerful information that we could then have. And as I say, there's a very wide range of mass spectrometry imaging methods. These are the ones I established the National Centre in the UK, and we have these MALDI, DESI, the different types of SIMS technology, there's secondary ion mass spectrometry, laser ablation ICP-MS, and even techniques that you can use in surgery while you're cutting through tissue, and you can get a mass spectrometry readout. But I'm going to be talking today about this particular technique called OrbiSIMS, which I developed at MPL. So in this slide here, I just wanted to give you a very quick overview of the kinds of things that we can do with mass spectrometry imaging. We can look at, for example, here, different metabolites that are involved in cancer. We can zoom in at cellular detail. I'm going to show more examples of that. Looking at different metabolite changes or looking at neurotransmitters. These we can see in here, for example, dopamine and GABA. We can look at things on the surface of cells. This is a paper we published in Nature, led by the Francis Crick Institute, where we could localize the artificial sweetener, sucralose, onto the cell membrane, and that allowed the biologist to understand what the mechanism of action was, because this artificial sweetener suppresses the immune system. It reduces the T cell population. I'm going to show you some examples later of organic electronics. This is a so-called depth profile. We go down the depth of the material and look at the chemistry. And yeah, more imaging applications. So it's a very powerful technique used in many different applications. And this slide really summarizes the whole field of secondary ion mass spectrometry. This is the fundamental process that goes on. We fire ions in at the surface. We set in motion molecules below the atoms and molecules below the substrate. But some of that energy comes back to the surface, and then we desorb atoms and molecules. And these days we can desorb actually quite large intact proteins into the gas phase. These are then detected in a kind of, whichever mass spectrometry you prefer, from quadrupole, magnetic sector, time of flight, or Fourier transform instruments. And we can use a probe beam, either single atoms. The single atoms like this we would use in semiconductor applications, where we want to get the ultimate spatial depth resolution of a device. So every semiconductor company around the world, every semiconductor fabrication plant has a SIMS instrument, and they will use this, and they can get a depth resolution of less than 1 nanometer per decade change of intensity, and from that they'll work out what boron dopant distributions are and things like that. It's a very important technique in the semiconductor world. Then we can increase the size of these clusters, and we do that because we want to cause less damage at the surface until we get to these very large argon gas clusters or water clusters, which are very much gentler and causing minimal degradation to the material. So just very briefly for those who don't know about SIMS, I just wanted to give you a quick evolutionary diagram. So this is kind of what you would look in your biology textbook. So these are epochs of time, and this is the sort of pathways that things grow, and then from Darwinian selection they grow and die. We have something similar in the SIMS field. It's actually been going many years. Each of these lines is now a decade, and you can trace its history, right back to the early 50s. In those times, really the work was pushed and developed for the semiconductor industry, or the emerging transistors, because they wanted to look at these boron or phosphorus dopant profiles. And this led to an evolutionary branch, which is called dynamic SIMS. This grew very large and is now not so much research in that area because they've perfected everything very well, but it's used, as I said, in every semiconductor fabrication plant worldwide. But another branch formed off, led by Professor Alfred Benninghoven in the 70s. And this was really steered towards looking at biology and organics. But as I described, you get this Darwinian process, so the kind of mass spectrometers they used at the time were not really very good for this application. And then there was development of new mass spectrometers, so-called time-of-flight mass spectrometers, which could detect all the molecules kind of in one go. But the downside of these time-of-flight mass spectrometers, as we'll see later, is they're not very accurate in terms of identifying what the molecule is, because their peaks are very broad and they're not very accurately located. Then there was a big period of ion beam development. So we've generally had these epochs of mass spectrometry development, ion beam development, mass spec development, ion beam development. But currently we're in a big period of mass spectrometry development. And I'm going to talk about this method here, which I developed, called OrbiSIMS, which again, going back to what I was saying at the beginning, is we're trying to get the highest level of confidence on what that molecule is and exactly where it is. And that's why I developed this new technology. There are now many of them around the world, as we'll see. So this is actually the reason, the physical reason, why we have this uncertainty principle. Don't worry about the details on the slide, but on this axis here, we have the amount of fragmentation that's caused by the ion beam in the mass spectrum. And on this axis, we have the spatial resolution that we can achieve in the beam. So this is a kind of a measure of the uncertainty in the identity of the molecule. If we have a lot of fragmentation, then we obviously don't have the original molecule, so our uncertainty is very poor. And yeah, we have a very high uncertainty here. But the types of ion beams that we use that can achieve very high spatial resolutions, so down at about 50 nanometers, unfortunately cause a lot of fragmentation and hence have really we have lack of confidence in what that molecule is. At the other end of this extreme, we have techniques which have a very broad beam, but they cause very little fragmentation. So now we know exactly, what that molecule is because we haven't fragmented it, but we've got very poor spatial resolution, kind of hundreds of microns. So everything I'm trying to do is trying to sort of find how far can we push this curve down to get better spatial resolution and better confidence in the identity. So this is the Heisenberg uncertainty principle. And then this is effectively what I'm saying in our molecular uncertainty principle, that the uncertainty in the spatial resolution is inversely proportional to the uncertainty that we have in its identity. And this is the big problem. It's why we have this reciprocal curve here. So how do we try and get to these optimum conditions? And one of the big advances in the field has been this move away from small elemental ion beams to these very large clusters. And this is a slide I've taken from a good friend of mine, Arno Del Corti. He's a specialist in molecular dynamics simulations at the University Catholique Louvain in Belgium. So what we have here is he's modeled glucose oxidase on a substrate on a surface here. And then he's firing in, this is an argon cluster. So it's many thousands of argon atoms that have coalesced together. And if I fire the movie, what you'll see is this molecule coming down and hitting the surface. Obviously, this is not in real time. This all happens in a picosecond time interval. So you can see now this gas cluster has disintegrated. Some of the energy has gone to the surface, but because all of this energy is shared out amongst many thousands of argon atoms, not much of that energy goes below the surface, and the overall energy that's spread out is quite low. We can entrain these protein molecules in this emission going out, and then we can liberate surface molecules into the gas phase. And in this case, we've got a whole protein molecule coming off, which is not fragmented. So this is very exciting because remember, we want to get confidence in the identification of the molecule, and we can only have confidence in the identification if we can keep this molecule intact. So one of the key things of developing a new kind of instrument was that we had to be very good at using these kinds of ion beams, these gas cluster ion beams. Just over 10 years ago, I was very fortunate to meet a famous clinical pharmacologist, Sir Colin Dollery, and Colin was very passionate about making better drugs. And how do you make better drugs? Well, most drugs these days, he worked for the pharmaceutical company GlaxoSmithKline. Most drugs, if they fail not because the drug is not because it's not safe, because the pharmaceutical companies now are very good at screening out toxicology type things. But they normally fail because they just have a lack of efficacy. And that's a real problem because the drug development process is something that goes on for about 10 years, from the beginning of having a particular biological target you want to try and treat to having a medicine at the other end. And the whole process of taking a drug from one end to the other, it costs something of the order of about $3 billion US, that kind of amount. And as we go through this process, the cost starts racking up. So once we get into animal studies, these preclinical studies, then you're spending hundreds of millions of dollars per set of experiments. When you get into phase 1, phase 2, phase 3 clinical trials with humans, you're spending $500 million. So the question is, can we spot early warning signals very early on as if that drug might fail? And this is when Colin came to my lab because he knew I was doing this mass spectrometry imaging, and we started to collaborate together to see if we could do that. And the instruments at the time, these time-of-flight SIMS instruments, showed some early ability to be able to do that, but actually we couldn't get confident identification of what the molecule was at the same time as getting enough spatial resolution. But luckily I had an idea for a completely new, different type of instrument. So this is really essentially what we're looking at. We're looking at a cell which is about 30 microns in size. This is obviously the nucleus here. Then you have all these different organelles like the mitochondrion, or you have lysosomes. Oftentimes drugs can end up going in the lysosomes, and then if they end up in these organelles, that can cause toxicity. So we really want to know if the drug gets to its biological target, the intended place where we know it will have action. So the pharmaceutical company, Pfizer in particular, developed this kind of framework to try and improve this production process of getting a drug from the beginning to the end more effectively, and it's called the Pfizer 4 pillars. So if we imagine we take an oral medicine here, and maybe this medicine is designed to treat these epithelial cells in the intestine. Let's say something's gone wrong and we have a biological target in one of those cells that we want to get our drug to. The question is then, the first pillar is, do we get our drug to the biological target? So this is why we need to have an imaging method that can go down to below 30 microns in resolution, and to be able to very accurately identify that drug in that location. The next kind of pillar is, does this drug bind with its biological target? So the target's normally a protein. Many of the drugs these days have intracellular targets, so we need to measure the drug inside the cell. Remember I was saying about the lack of efficacy in some drugs. Well, this is a big drive in the industry now, is to do this tests in human tissue as soon as possible, because obviously there's a big difference between rat and mouse and human. So the sooner you can get data from human studies, the better you can understand if the biology is really being effective. Well, mass spectrometry imaging is very good at looking at things from human tissue. We can take very small samples from biopsies, and we can analyze that in our instruments very well. So we're really well configured for looking at this so-called functional pharmacology. Can we see the changes in metabolism, the chemistry of life as the drug has been taken up? Because if we can see these metabolic changes, then we got a good indication that the drug is being effective. And the last point is, are we looking in the right phenotype, a relevant phenotype? So again, we're doing it in human and in populations of the human which are known to either be treatable or not treatable. So a lot of work now combines this with single-cell genomics and single-cell transcriptomics, and increasingly single-cell proteomics now. And what we're doing is we're overlaying this metabolite information in this multi-omics picture of where the drug is. So let me go back to this kind of, we now know we need to have these gas cluster ion beams like I showed in the movie to get low fragmentation. What do we need from our mass spectrometer? Well, the mass spectrometer has got to be able to resolve the complex chemistry that we have in a cell. This is an example of the mass spectrum from two different mass spectrometers of the same sample. This is using something called a Fourier transform ion cyclotron resonance mass spectrometer. And here you can see we can completely resolve these two different molecules. But if we use a time-of-flight mass spectrometer, we can no longer resolve this. This is completely blurred. And that is a problem. Now, there's a good reason why we use time-of-flight mass spectrometers in SIMS, and it's because of this plot here that I've overlaid. And on this axis here is the time it takes to take a spectrum. And on this axis here is the mass accuracy that we can get from this technique. So this is how accurate is the position of that peak on the mass scale. This is critical information to identify the molecule. And here I've converted this time per spectrum to the time for an image, because we're normally doing this in an image. And you'll see what happens. These Fourier transform instruments are fantastic in terms of having very high mass resolution and very high mass accuracy, but they're very slow. So they take many, many hours to take an image, which is too slow in most cases. Time of flight mass spectrometers like OrbiSIMS, we can effectively, in theory, we can take an image in seconds, although we normally take minutes because we need to get enough signal to noise. And then on this, I've added in several different types of mass spectrometer. But my idea here was that fundamentally, we just can't have a mass spectrometer which gives us this kind of level of mass accuracy, but at the same time is really fast. There is nothing that gets down to this region here. So my idea is, why don't we combine in a dual analyzer configuration a time of flight mass spectrometer and an Orbitrap mass spectrometer into one instrument. And then by having these two mass spectrometers, we can get the best of both worlds. And this is a similar kind of plot, but looking at the mass accuracy, so the mass resolving power. So this is how sharp the peak is. 10,000 is a typical number that you get from a OrbiSIMS instrument. But these Orbitrap mass spectrometers, which is a kind of electrostatic Fourier transform mass spectrometer, can get 500,000, or even we can get 1 million these days in terms of the mass resolving power. Fourier transform instruments have even higher. They can get up to 10 million, but again, are very slow. So now we have the ability to do imaging fast with the time of flight mass spectrometer, but then we can use the mass resolving power of our Orbitrap to get then these very sharp identifications of the peaks. Anyway, that was the idea, and we were lucky enough to get the funding to get this multidisciplinary project off the ground. And it involved collaborating with IonTOF, who you will know well, who developed the OrbiSIMS here, with Alexander Makarov, who's here, who developed the Orbitrap technology at Thermo. And the two companies then worked together with my colleagues at GSK. This is Sir Colin Dollery, and this is Sir Keith Peters, a very famous clinician. They worked together to build this new instrument based on my design. And yeah, this was the first one in November 2016. It's still running in my lab. And yeah, this was the beginning of really a revolution in the field of SIMS. So this is, we published the paper in Nature Methods in 2017. And this schematic kind of tells you all the things that you kind of need to know about this technique. We have the time of flight mass spectrometer. We have the Orbitrap mass spectrometer. We also have different ion beams, so the gas cluster ion beam for this very delicate analysis of biological materials. But we also have ion beams for looking at semiconductor materials, these small atom-type beams. And we can use it for getting spectra, we can use it for getting depth profiling, we can use it for imaging, and we can use it for 3D imaging. So the postdoc in my group at the time was Melissa Passarelli, and she did a really great job in this work. She's now at Concordia University in Canada. As part of this project, we recruited Alexander Perkel into the IonTOF company. He had experience in Orbitrap mass spectrometers from his work in Gießen University. And recently, last year, he got an award from the American Vacuum Society, which I was really pleased to see for the amazing work he did in engineering these instruments, because there are now many of these around the world. There's over 20. We just got the 20th one in my lab at the beginning of this year. And IonTOF are making these, I think, as fast as they can sell them at the moment. They're very popular. So from that Nature Methods paper, this slide really brings into a very good example of this whole kind of concept of knowing where something is and knowing what it is exactly. So with the TOF mass spectrometer, the time of flight mass spectrometer, we can image tissue very fast. So we can take an image of this mouse brain in a matter of minutes to do a quick survey. But then what we can do is we can switch to the Orbitrap mass spectrometer, because let's say we've identified a region. This is an H&E pathology slide. We say we want to go and have a look at this particular region to see what's going on. And now we use the Orbitrap with the gas cluster ion beam, and now we have a really high resolution, just over a micron spatial resolution, combined with beautiful mass spectrometry capability. So with this, we could identify uniquely more than 70 different, oh, it's maybe my mobile phone. I'll take it out of my pocket. It's interfering, maybe. Yeah, we identified over 70 different lipids. You can't see too well from this picture here, but this is where the neurotransmitters are, like dopamine, GABA, serotonin. These are actually a very low-level neurotransmitters, and what we were able to do is uniquely identify those. And this is where you have to have this very high mass resolving power in your instrument to separate out all of these biological peaks. And we were able to show that previous literature using OrbiSIMS data, they were just blurring all the peaks together, and dopamine, for example, is a very low intensity peak, it's a low abundance peak, but we could identify it uniquely. But we could zoom in right into an individual cell and then look at the nucleus of the cell, and then we can look at the chemistry within the nucleus of the cell. So this was going from the tissue to the cellular to the subcellular scale. So Colin, as I said, was a clinical pharmacologist and not a mass spectrometry person per se, but so it's very nice when you collaborate with someone, they have a different point of view. And he used to draw his mass spectra like this, upside down, because it was easier to read the labels. But what we could do in this mass spectrometer now is you can see how sharp these peaks are, but we could pick a particular lipid, like this one here, and then we could fragment it, and we could get the MSMS spectrum, and then look at all the fragmentation products on that, and then look at the structure of that particular molecule. And we could also look at individual cells. So in this case, we took rat alveoli macrophage cells and we incubated them with the drug amiodarone. Amiodarone is a drug for cardiac arrhythmia, but it has not a very nice side effect, that it causes something called phospholipidosis. It causes the macrophages to go foamy. This is because the drug gets trapped in the lysosomes of the cells, and then the cell then forms a protective layer by growing multi-layer lamella around the lysosome and generating this phospholipidosis state. So what we were interested to do is see if we could see the drug in these lysosomes, and could we find a biological biomarker which is, instead of trying to identify what's going on using electron microscopy, people looking at these structural effects, could we just get a biological marker to identify this phospholipidosis? So within this, we could take individual cells from a population of cells, and we could look then at the drug, which is the green molecule here within the cell. But what was very interesting is that we were able to then measure cell by cell the amount of drug uptake. So this is the amiodarone uptake. This is the expression of a particular lipid, the phosphocholine 34.1 lipid. And you can see if we put the data for all of these different cells on one plot, they're falling on the same line, but some cells take up a huge amount of amiodarone. Some cells hardly take up any amiodarone. But the cells, you can see there's a fairly good correlation between the amount of drug uptake and the expression of this phosphocholine lipid. So we could identify this particular biomarker. But this was really, really early in this period of people looking at single cell heterogeneity, and that some cells behave, even though they're from the same cell line, are behaving completely differently in terms of drug uptake. So this was, for biologists, this was really interesting information. So as I said, now there are many OrbiSIMS around the world. This map shows, I mean, it's kind of, there isn't one in Hawaii. It would be very nice if it was in Hawaii, but Google Maps just colors the whole territory. But yeah, it's an ever-expanding community. And from our original Nature Methods paper in 2017, there are now really many, many OrbiSIMS papers. And on this slide, I've just put a selection of the ones that have ended up being published in the wider readership journals. I mean, not to say these papers are any better than any others, but just because they're solving problems that you probably couldn't do with another technique. They've ended up in journals that are kind of wider readership. Some of them are from my group and some of them are from other groups. This is a collaboration with the Francis Crick Institute. This is really our first sort of cryo OrbiSIMS data. This is from the University of Nottingham in PNAS. Completely different application, looking at archaeological clays and ceramics. But they use this OrbiSIMS, the power of being able to identify the chemistry and know exactly where it is to look at these things. Protein identification from Gustavo Trindade, who's now in my group, and the folks at Nottingham. You could then identify the protein layers in 3 dimensions as you go through the skin. This is looking at mesenchymal stem cells, so a biological application. Again, looking at skin, as our paper with the Francis Crick Institute, where we were looking at the artificial sweetener sucralose on cells. More recently, we did this paper in collaboration with Samsung, published in Nature Communications. I'll show you a bit more about that later. This was a very nice publication recently from the University of Nottingham, where they used the OrbiSIMS to constrain computational methods, but allow you to get the 3-dimensional structure of RNA in its bound and non-bound states, which for me was amazing. And there's been several other very exciting papers that have come out since I did this slide. So one of the big advances for us, especially for biology, was moving into cryogenic analysis. And this was the work of Junting Zhang, who was in my group, a postdoc in my group. She's now in a pharmaceutical company in China. And she was a heroic worker, because this is very difficult to have everything running on the instrument and then get the cryogenic sample preparation correct. So we obviously learned a lot from the cryo-EM community, the Nobel Prize, to the Richard Henderson at the LMB in Cambridge and the others. They really pioneered, of course, the use of cryo-EM. And we've taken the same sample preparation methods that they use, high pressure freezing technology, the ability to move the sample and position it into our instrument without it ever seeing air. But Junting had to find methods that were compatible with mass spectrometry. The EM protocols just wouldn't work with mass spectrometry. They killed our ionization process. But she was able to, she pioneered and she managed to do it. And this is the project that we had at the time, was to look at Pseudomonas biofilms. So Pseudomonas is a nasty bacteria which is involved in hospital-acquired infections, for example. And it forms a very nice and complex biofilm. These biofilms protect the bacteria from drugs, which is a problem. So if you're trying to kill the bacteria, you've got to get through this kind of protective scaffold. And it's composed of many different things. There's water, so this is why we need to do cryo, because our systems are vacuum-based. If we put water samples in, it obviously evaporate. They have polysaccharides in, they have a DNA which comes out of the bacteria and goes into the extracellular matrix. There's proteins and these quorum sensing signals. So it's a really complex architecture. And what she was then able to do was to high pressure freeze these films, then do high resolution TOF-SIMS imaging, but in the OrbiSIMS using the TOF analyzer. But at the same time, so this is the 3-dimensional image from the TOF analyzer, looking just at potassium, which is a marker for where the bacteria are. But at the same time, we're acquiring data with the Orbitrap mass spectrometer. So you can see here we can uniquely identify what the different peaks are. But we can also pull up the images from the Orbitrap, and with that we could then identify these quorum sensing molecules. This is going by layer as we cut through the biofilm. We can see then these quorum sensing molecules is how the bacteria communicate. And we published this data back in 2020. Then more recently, Anna Kotowska at the University of Nottingham took that data set again, and she developed some very nice computational methods to mine the data. So it's the same data set, but now she's able to look at these depth profiles going through the material and identify a whole range of different polysaccharides, quinolones, protein fragments. And this is the substrate. So it's a very rich kind of data set. It's the kind of data you would have in liquid chromatography, mass spectrometry type experiments. So as I say, I collaborate a great deal with the Francis Crick Institute. My biological collaborator there is Alex Gould, who's an amazing geneticist. And we had a student, Claire Newell. So she applied OrbiSIMS. The great things with Drosophila is, of course, that we can completely manipulate the genetics in that to change the metabolites that are on there. Now, I'm not going to go into the biological questions that we're trying to solve, that Alex is trying to solve, but with Drosophila, they have models, for example, for Alzheimer's, they have models for Parkinson's, all sorts of different, for cancer. So we're really interested to see if we can map out the metabolism. So in Claire's PhD, she was applying cryogenic OrbiSIMS to look at the cuticular surface of these fruit flies. And this just shows you the level of control that we can have. So this is a control Drosophila here. And this is one where we've knocked out a particular gene, and then we follow then the metabolite chemistry at the surfaces. And you can see in the control, then we can see this particular metabolite, but when we go with the knockout, it's completely disappeared. And we can do that, then we can knock things out, and then we can follow the metabolic processes that occur down at the cellular scale. By going to cryogenic conditions, we do bring advantages for the technique. One of the things is we reduce fragmentation. So this is room temperature, and this is cryogenically. And by going up in this direction, we're reducing the amount of fragmentation. The other thing is the water matrix. In this publication here, we found the water matrix enhances the ionization of polar molecules. This is very important. Many drugs, for example, are polar molecules to get good solubility. So the more, if we can have high ion yields, we can get better sensitivity. So cryoanalysis is a really important topic for us. And Alex and I were fortunate to get a Wellcome Trust grant to develop the OrbiSIMS into the sort of the next kind of level of imaging. At the beginning of the grant, this is the kind of images that we could get from Drosophila. This is the so-called neuropil in the center, and this is the cortex. But now we can get much better images. So now we've moved on to using not argon clusters anymore, but CO2 clusters. So this is the work of Claire Newell, who's now a principal laboratory scientist at the Crick, with my two PhD students, Anya Ayres and Yuhong Jin, who's also one of Alex's PhD students. So what we can now do is, we can take cross-sections of the Drosophila CNS. This is the neuropil, the cortex, and now you can really start to we're seeing the detail of these cellular structures here. We can follow different changes in the Drosophila if we change its diet or we genetically change it. And we're now pushing this technology to go even higher resolution. So our aim is to get to submicron resolution and get the complete 3D molecular image of the brain. I'm going to skip that one. So let me switch a little bit now to material science applications. And this is an example where we used OrbiSIMS to look at an organic electronic device. So if you have a phone like this, it's probably an organic electronic display. And if you look at one particular pixel, you'll see that it's a very complex organic layered chemistry with whole transport layers, electron injection layers, barrier layers, all of these things to make the device work. The thickness of this is only a few hundred nanometers. So it's very complex chemistry in a few hundred nanometers. The blue LED, the blue color is the hardest to make stable. And this is still been a big challenge in the OLED industry. And you often see papers in Nature Communications about new advances to improve the lifetime. So we worked with Samsung to see if we could find what was causing the problem. Why were these devices failing? So this is an OrbiSIMS depth profile through one of these devices. Every single pixel, every dot you see here is a rich mass spectrum. So we're getting all of that molecular information from less than 1 nanometer of material. So very incredible level of information. And the resolution that we get is very high, so we have the ability to accurately identify what the chemistry is and very accurately localize where that chemistry is. And because of that, we could identify, this is as we start to age the device, this is a fresh device, there's a 5% aging, 30% aging. We could start then to see the evolution of certain molecules. These are sort of attomole levels of concentration, but because our technique was so sensitive, we could identify what they were. We could see how they correlated with increasing degradation of the device. But then once Samsung knew what these degradation molecules were, they could change the host chemistry, and then by changing the host chemistry, they could improve the lifetime of the devices by a factor of 10. So this is a really nice example. Samsung actually have 2 OrbiSIMS running all the time for these kinds of applications. Another application in materials science is in the semiconductor industry, which, to be honest, I was not expecting semiconductor industry to be interested in OrbiSIMS, but it turns out they are. And the reason is because we have this very high mass resolving power. So as you'll know, devices in the semiconductor industry have got smaller and smaller and smaller. They're now at like a 3 nanometer node, much smaller than the spatial resolution than you can get from chemical probes. But by looking at co-emission of things in the mass spectrum, we can tell whereabouts it came from. So for example, in this device here, you have silicon dioxide, you have germanium, phosphorus, germanium phosphide. If we look at the time of flight sim spectrum, which is the red curve, so this is the work from IMEC, by the way, Alexander Franket and colleagues at IMEC, it's not from my group. They then took data, this is the TOF data, overlaid is the OrbiSIMS data. There's 2 things to take note of here. One of them is that obviously the Orbitrap data has got much, we can separate out all of these different peaks now. So we can separate out germanium phosphide from germanium oxide. By doing that, we can then do a depth profile from just those particular peaks, and then you don't have confounding chemistry, and that's how they can then get their quantification. This is a method that they call self-focusing SIMS. Another application that they used it for was in extreme UV lithography. So extreme UV lithography is what they use now to get these 3 nanometer and below resolution, and very, very fancy technologies that they use to do that. And the photoresists have got different chemistries. So they used OrbiSIMS to identify what was happening. With TOF SIMS, the peaks were too blurred. They couldn't identify what was going on. But by switching to OrbiSIMS, they were then able to separate out the chemistry and figure out what was going on. So we were really interested then in better understanding how could we improve the technology for the semiconductor community. So in a collaboration with IMEC, we looked at a standard sample, which is antimony delta layers in a silicon matrix. So this is work of Yundong Zhou in my group. This is a depth profile now, so we're going down in depth to a few hundred nanometers, and we're measuring now the amount of antimony atoms that have been implanted in a silicon wafer. And we compared our Orbitrap technique with a magnetic sector instrument and a time of flight analyzer. I don't really have time to go into the details, but basically we can, because we have such high mass resolution, we don't pick up some artifacts that you get from other techniques. But the key parameter here is something called the useful yield, and this is our OrbiSIMS, this is the TOF SIMS. I should say TOF SIMS is a brilliant technique, don't get me wrong, it's an amazing technique. This is a magnetic sector instrument that they use in the semiconductor industry, but we get the sensitivity of our instrument here is pretty much the same as these dedicated magnetic sector instruments because of the way our instrument works. Okay, in the last few slides, I want to talk a little bit about some of our most recent work. This slide was inspired. Roger Penrose is a very good friend of my wife, and she's with Roger today, actually, and mine. And you probably know Roger has this impossible triangle concept. Well, people in the metrology world are obsessed with triangles, with so-called metrology triangles. We're always trying to unite things together to triangulate and make measurements more accurate. And we had a problem that in OrbiSIMS, It's not a single ion counting system like in a time of flight mass spectrometer. So we just have an analog detection of the signal here. It's kind of uncalibrated, basically. So we can't link signal, noise, and transmission together uniquely. So it's an impossible triangle. So in single ion detection, which we have in your time of flight analyzer here, is very nice because in this example here, the apple falls on Newton's head and Newton goes, one apple, I detected it. But it's not like that in an Orbitrap. So in an Orbitrap, the ions are cycling round and round and round and round and round, and we detect an image charge, and that image charge is in this operational amplifier here. We get what's called a transient, and then we do a fast Fourier transform. This is actually a very complex process, and we've been collaborating with Mike Keenan, who's retired now, but was a statistician leading an analytical chemistry group in Sandia. For many years, we've been working out all of the instrumental aspects that lead to noise in the instrument. And I don't have time to go into the detail here, but it is complicated. You end up with something called a Riccian distribution, but there's a lot of signal processing that goes on to reduce the inflation in the mass spectrum just caused by noise alone. And it's a kind of complicated function, and as I say, I won't go into the details here because, well, it's really probably not too much interesting to you, but there are two very important things that came out of this. One of them is we could now calibrate our intensity scale because this portion of the curve here is Poisson. And because it's Poisson, then we can then work out how many ions were there. So we now calibrate our mass scale so that now our impossible triangle has now become possible. But if you, in our field, in mass spectrometry generally, the data is so complex that people use multivariate analytical techniques. But the data in mass spectrometry, in all mass spectrometry techniques, is what's called heteroscedastic. So that means the noise is not uniform across the mass scale, it varies. So a peak with high intensity has a naturally high amount of noise compared to a weak peak. Multivariate techniques like principal component analysis resort out the data in terms of variance. So it will then be biased to high intensity peaks, which just have naturally a high amount of variance because of the counting statistics. These weak peaks, which actually are probably the analytically important peaks, for your drug molecule, for instance, will then be kind of discarded and they will be left behind. So you need to account for this. So this is actually our theoretical measurement of the noise in the instrument. And this plot here shows how if you use no scaling in multivariate analysis, it biases the analysis. It's only by doing it using our method, the red curve, that you get unbiased data. And well, we won't go into the details here, but it basically means by doing the correct statistical scaling, you get much more efficient use of multivariate analysis. This is a paper that's currently in revision. We're anxiously waiting news from the second round with the reviewers. So the other important point of this is that we have a noise floor in the Orbitrap. So this is this semiconductor material I showed you earlier, now plotted on a calibrated intensity scale. And you can see it runs out here because the instrument software cuts out the signal, because here it's just completely noise. We're totally swamped with noise. So this is this bit here. This is a Riccian distribution, would really go like this. But here, the computer just cut out all of the signal. So we're losing signal. And here our signal is mixed with noise. So when I saw this, I thought to myself, well, why do we just use an analog detector? Why don't we use a quantum detector? So this led into a project and a collaboration with colleagues at MPL, John Gallup and Ling Hao, who are experts in quantum detection. And then we patented a quantum detection system. The very impressive thing about, and the reason why we have the patent, is that our detector system works in a broadband mode. So we get single ion detection across the entire mass scale, because normally squids are sensitive to one particular frequency, which is fine, but a mass spectrum is more than one peak. We need to do it across the whole mass spectrum. So we've been developing a proof of concept system. We now have This is a test bed system. This was part of the OrbiTrap mass spectrometer in our original quantum detector. We now actually have it, we now have an OrbiTrap mass spectrometer, the complete system. We're able to do proteomics in the quantum lab. And very soon now, we should be getting the first results with our quantum detector. So we're very excited about this because there are thousands and thousands of OrbiTraps around the world, probably 10,000, I think, used for proteomics, metabolomics. And if we can then improve their limit of detection by a factor of 10, I think that's going to be really important. So yeah, coming to my conclusions, new SIMS instrumentation has revolutionized capabilities for biological and material sciences. And when I mean new SIMS instruments, I mean the instruments that you have here. You have a really state-of-the-art TOF SIMS instrument. It's very well equipped. And you have really, I've been so impressed this week seeing the expertise in the people here and how much you know about the SIMS details. So congratulations to the team here, because that's impressive. It's kind of a complicated technique to use, and to have learned so much in such a short time is really very impressive. The OrbiSIMS enables simultaneous localization and identification with high confidence. We can do it to the nanometer scale if we're looking layer by layer. For inorganic materials, we can do it to about 100 nanometers, and for organic biological materials, we can identify exactly what it is to around about a micron, submicron resolution. The cryo-OrbiSIMS allows native state imaging with enhanced sensitivity, and this is really, in my group now, is a big push for everything biologically we do is cryogenic. The quantum detector shows the promise to improve the dynamic range of OrbiTrap mass spectrometry by a factor of 10, and I think this is going to be very exciting for people, particularly in like single-cell proteomics and single-cell metabolomics, where sensitivity is a real challenge. And now, we took the 20th OrbiSIMS, was delivered to my lab in January, and I know Einthoff are making them as kind of fast as they can sell them these days. And they're all around the world, and kind of what surprised me is they've ended up in, remember I originally developed it for biological applications, but now it's used in semiconductors, organic electronics, pharmaceutical, massive range of different technologies. So it's really, for me, continues to be amazing. We have now a very nice community of OrbiSIMS users, and more details are on our website. I'd just like to thank my team, it's an amazing team, and all the people who've been in the past members of my group that have now gone on to many different brilliant positions. And also particularly like to thank Alex Gould, a Fellow of the Royal Society in the Academy of Medical Sciences. Amazing collaborator, and we have a very nice biological collaboration together. And with that, I'd like to thank you very much for your attention, and happy to hopefully answer your questions.
Open to questions, comments.
Thank you for the talk. Very nice. Can you comment a little bit more about this superconducting kind of squid, I believe, and bunch top stuff?
Yeah. Yes, the answer to that is yes and no.
Okay.
See, no. Because of the patents, there's a bit limited to what I can tell you. Because we're not giving out too much detail about the actual configuration. This is an OrbiTrap here, outside of the vacuum system, and this was our original test system that we had running. We now have it actually connected up to an OrbiTrap mass spectrometer, the complete system. The key advance that we did compared to the rest of the community, the prior art, let's say, was our system can work across a complete range of frequencies, and that's the kind of the novel bit. But the details, unfortunately, I'm afraid I can't give now.
Okay, but it's not that you are cooling down
no,
something
this bit
down
we
there?
don't cool down. No, in the actual device So this is the actual trap here, but the superconducting device is held away from that in a cryostat.
Okay.
At the moment, we have to do it sub-cell 7 Kelvin, I have to say, because of the types of superconductors we use. But this is obviously not very convenient for mass spectrometry labs, but our intention is to develop a higher temperature liquid nitrogen version in the future. Just make it easier for people to use.
Okay, thank you.
Thank you.
Hi, Ian.
Hi.
I have a question regarding the pharmaceutical industry.
Yes.
You told us a process that Pfizer developed in order to assess the effect of the drugs that they are manufacturing, or not manufacturing, like developing.
Yeah.
My question was, how is the technique, the SIMS technique, how involved is the SIMS technique in the regulatory affairs? I mean, if it's an essential essay that they have to do, there are other essays, of course. My question was, how is the SIMS technique involved in that kind of approval and regulation?
Oh, that's a really good question. Thank you very much. Okay, so for drug discovery, so far it's not been part of any regulatory submissions, because it's kind of used as part of the tools that they use to test and develop. But there has been work with the FDA, and I think there's increasing likelihood for some applications, sort of topological treatments of skin, because SIMS, as you will know here, is very good at analyzing skin and seeing where the drug goes within the skin. And increasingly, there's work going on with the FDA, I think, to actually make that part of submissions. I think it is probably something that's coming, I think, particularly with the sort of the more expertise now. Pharmaceutical companies are using SIMS a lot more than they used to. So it is becoming part of their normal processes. Where it is, I think, part of the QA type procedures is in the packaging side. So SIMS is extremely good at looking to define the chemistry that's involved in the packaging, and also to find if there are problems. So I think probably from a regulatory perspective, it will be packaging first, then it will be sort of topological applications to skin, and then probably more in the sort of general R&D side. But yeah, really good question. Thank you. Thanks.
More question? Okay, we thank again Professor Gilmore.